continuing with our cutaneous membrane and accessory structures, which we call our integument or integumentary system. We talked about how the epidermis and dermis made up the integument, and the accessory structures were things we were pointing out, like our sebaceous glands, our nerve endings, our hair follicles, our sweat glands, even our blood vessels, and all that actually makes up the actual structure of the skin. And of course, we also have what we call subcutaneous fat in here, and then we have areolar tissue that's connecting the epidermis to the dermis. We even have some areolar tissue, some fascia that connects the skin to other things like our muscle or other areas of the body that the skin is connected to that don't necessarily have muscle, whether that be to bone or tendon or something like that. Okay, so anyway, we do see that our skin is actually an organ, and organs, remember, are made up of tissues. So the tissues that we see here are obviously epithelial tissue, because the epidermis is primarily epithelial tissue. It's connective tissue. There's a lot of connective tissues here. The dermis being our probably largest sample of tissue. Blood is a connective tissue. Fat is a connective tissue, but our nerve endings are actually part of our nervous tissue. And then so we have examples from epithelial, from several connective tissues. Nervous tissue is part of this, because obviously this goes back and attaches to our nerves. And even muscle tissue, because we have these things called erector pili muscles that basically cause us to get the goosebumps, or cause the hair to be able to stand on end, and actually have the ability of producing, as they contract, as all muscles do, producing a little bit of heat. And when we get cold, that's when we tend to get the goosebumps or the chill bumps, whatever you want to call them, because we're actually starting to produce a little bit of heat by contracting these muscles. Okay. So, looking at this upper section here of epithelial cells, let's look into that just a little bit deeper. Now, notice again, I've got this is my epidermis, which is epithelial. All the epithelial all the way down to this region right through here. And then my basement membrane is basically what I would be outlining through here, and the germ cells or the stem cells that would actually be in that section. And then of course I've got my dermis down here, which is again my irregular, dense, fibrous connective tissue that's vascular. Now, but let's concentrate just a little bit on these different layers of the epidermis and talk just very briefly about what's going on in these different layers. So the stratum basal, actually also sometimes called the stratum germinativum, that is our very first layer of cells next to the basement membranes. And its main job is it is where we contain our stem cells and our melanocytes, cells that produce melanin, but our stem cells, which are constantly going through mitosis. They're constantly dividing, okay, and they keep dividing up. They divide up even through the next layer, the stratum spinosum, several cell layers thick. The cell divisions keep dividing, and then we get up into this area called the stratum granulosum, and at the stratum granulosum, we actually start producing a bunch of a protein called keratin. So all these cells are alive and reproducing on up, through the next layers. They're close enough to the vascular areas here where they can receive all the contents of the blood. But after the stratum granulosum, then we have a very, and we don't find this in every skin layer, but a very thin layer called a stratum lucidum. And by the time the cells basically get up from the stratum granulosum to the stratum lucidum until finally the last layer, the thickest layer, the stratum corneum, the cells start actually dying as they produce the keratin. It starts crowding out the other parts of the cytoplasm, and the cells below it keep pushing against it, so they flatten. And by the time they reach our last layer, the stratum corneum, the cells are basically flattened and dead, but we keep pushing them up, and the outside cells keep wearing off. I think they said I read somewhere about 80% of household dust is dead skin cells that have been sloughed off or shed. So anyway, we shed our skin all the time. We just don't realize it usually, but we keep replacing it. And of course, as we age, that process becomes less and less efficient. We keep making copies of cells. It's kind of like if you take a paper and put it through a copier, and you keep reproducing the same copy from a copy from the next copy from the next copy, then later on your copies aren't quite as clear and precise as the original copies were. The ink and we start seeing little mistakes, and so in the DNA, as we age, that the ends of the chromosomes actually start wearing down a little bit, so we actually have an aging process. Not to mention that when you have things like ultraviolet light from the sun, different things that affect your skin, they can actually start affecting the division process and even affecting the consistency of the layers of the skin. But anyway, that's a whole nother story. But what I want you to see here is the kind of the progression of the cells from the cuboidal shape down here to the flattened squamous shape. But the reason why we usually just classify the whole thing as stratified squamous is because you can see the top layer here, the stratum corneum, is much thicker than the rest of this, but it's almost all simple squamous epithelial cells that are basically dead, because as they've gotten further away from their vascular area, then they die. Now, again, the epidermis is, if you peel, this is the part that usually peels. And of course, too much sun damage can cause that. We also see that we have cells in the bottom area called melanocytes, and they actually produce melanin. And if you have darker skin, you're producing more melanin, and the melanin is actually working its way up higher in the layers. If you're fair-skinned, you're not producing as much melanin, and the melanin that is actually living in little vesicles called melanosomes, they actually don't go as superficial, but they stay deeper. So if you're darker skin, you're much more protected from any ultraviolet light, because the melanin tends to protect the nucleus in each of the cells. If you're lighter skin, you're much more susceptible to skin cancers because you don't produce as much of this protective dark pigment that protects your nucleus. So you really, whether you're dark skin or light skin, if you're in the sun a lot, you probably need to wear some sort of sunscreen or sunblock. All right, I think these types of burns and how they rate them has changed a little bit, but we'll just go ahead and go with what this says. First degree burns being basically where something burns your skin enough to where it's red and swollen, but only the outer portion is damaged, like a light sunburn or a minor, pour some hot water on yourself that is just a minor burn. Now, second degree burns is where you not only damage the epidermis, the outer layer, but you get some of the upper dermis. And so you can usually tell this if you actually have blisters. If you have blisters, you have gotten down into the dermis area, so it's a second degree burn. And these are usually super painful. Okay, third degree burns is the worst of these three. Obviously, it's going to destroy all of the skin, both the epidermis and the dermis. And sometimes it will even burn the nerve endings. So it's usually a gray, white, or black. Now, I think they've got a different way of describing these now, and it may have actually a few more kind of subcategories. But anyway, those of you that are in the emergency medical service, I'm sure that's something you've gone over. But we'll just go with these since this is what the book has. Okay, let's talk about one of the most common cancers in the body, and that's skin cancer. And what cancer basically is, you see here, abnormal cell mass, but really it's when cells are dividing when they are not supposed to. So if you get cells dividing when they're not supposed to, then they're going to form some sort of abnormal mass or tumor. Now, we divide those masses up into two types, benign or malignant. If a mass is benign, it means that the cells are staying in one area and they're usually have connective tissue holding them in. So we say they're encapsulated. They have some sort of structure around them. The cells are dividing, but they're staying in one area. These are much safer because they're much easier to get rid of. But when we say that cancer is malignant, we say it is metastasized, or cells are breaking away from the tumor and are actually moving to other parts of the body and setting up what we call secondary tumors. And of course, these are the much more dangerous because as they spread, it's much harder to control. And as they spread, the proteins that create these malignant tumors, then if it's not caught early enough, then that's usually something that has a high mortality rate. So skin cancer is the most common type of cancer. So what I want to do right now is look at what we call the ABCD rule. And you can see A is asymmetry. They are not symmetrical. B, they have an irregular border. They have different coloration within the same area, and their diameter is usually larger than 6 millimeters, which is about like a pencil eraser. So looking at these particular, for instance, moles, and melanoma is the most dangerous of the skin cancers. So if you don't get anything else out of this lesson, please look at this, because this could be something that can save your life or save the life of somebody that you know. So the first thing are asymmetry. Notice how on our normal moles down through here, if you were to just divide it down the middle, one side is pretty much the same size as the other. If I divide that down the middle, one side's pretty much the same size. So divide it down the middle, one's pretty much the same. They're pretty much the same. But notice here, if I divide that down the middle, that side's quite a bit different than that side. If I divide that down the middle, this side's quite a bit different. Again, divide these down the middle. They are not symmetrical like these are. The next thing is the border. Notice how the border is smooth all the way around. Smooth, pretty smooth all the way around, smooth all the way around, and smooth all the way around. But look how that border is not smooth all the way around. It's kind of jagged and kind of, goes in different directions. Same thing here. Notice how the border is not just smooth all the way around, but kind of goes out in different directions. Okay, that color, notice how the color is pretty consistent all the way through. Same here, and same here. But notice how that's darker here, lighter here, lighter through here. Definitely darker here and lighter here. Darker in that area, lighter in that area. So if it varies, if the color varies instead of being pretty consistent throughout, then that's a problem sign. And then the diameter, notice these are within a certain range where all these are much bigger. So if you have these characteristics on any moles, then that is something you definitely need to get checked out as soon as possible, because melanoma cancers do spread and they do actually have a pretty high mortality rate. So the earlier you detect that, because it's very easily solved if you detect it, we can get those removed before they have a chance to spread their cells to other areas. So that's something that you need to look at. Another thing I like to also bring up is especially for those of you who are taking care of patients, especially older patients, is something that is called a decubitus ulcer, or commonly known as a bed sore. And this is caused when cells aren't getting the blood supply, because the blood supply is restricted, usually because they're laying in one place, or because they are dragged or pulled over certain areas repeatedly. So you want to definitely check your patients and make sure that they're not forming these ulcers because they're not getting enough circulation, again caused by the either not being turned or moved, or they're dragged over these pressure points repeatedly. So make sure you're taking care of your people or looking after your own relatives who may be in some sort of assisted living situation.